Welcome back to From Our Neurons to Yours from the Tsai Neurosciences Institute at Stanford University, bringing you, as always, to the frontiers of neuroscience. Today, we're going to be talking about healthy brain aging. And to do that, I want to introduce you to somebody named Harry Tu. Harry was born in Shanghai and spent his career as a coal mining engineer. He's now in his mid-90s, has no medical complaints, doesn't take any medication. He swims laps 5 times a week, carries his bike upstairs to his apartment after a long bike ride, and he's the one who drives around to visit his family and friends, most of whom can't drive any longer. And he flies out to China every year to visit his relatives. Harry is the picture of healthy aging. He's independent, he's able to live his life and do the things that he enjoys. He's part of a study at Stanford, the Stanford Aging and Memory Study, or SAMS, which is trying to understand what it is that allows some people to live into old age with their mental faculties largely intact, to live a fundamentally independent life. So the SAMS study has been running for more than 10 years, and it's led by Anthony Wagner in the Department of Psychology and Beth Mormino in the Department of Neurology, and supported by the Knight Iniative for Brain Resilience. I asked Anthony and Beth on the show to tell us what we're learning about how we can all age more like Harry Tu. And I started off by asking Anthony, how did SAMS start and how has it grown since then?
So SAMS started back in 2014. The initial idea is we were going to enroll a large sample of cognitively healthy older adults. These are individuals who, through extensive series of cognitive testing, appear to have entirely normal, intact cognition. We've enrolled now, well over 200 participants. And the idea at that time was to get just sort of this one snapshot of their cognition and to try to understand how variability in that cognition, both moment to moment, as well as between the different older adults who were enrolled in SAMS, how that variability relates to brain structure, brain function, to other biomarkers that are sort of risk factors for Alzheimer's disease, such as amyloid and tau proteins that at the time you could pick up in cerebrospinal fluid, but as SAMS and as science has evolved over time, you can now pick up with things like blood plasma. And of course, there are also other imaging methods like positron emission tomography, which we've added to SAMS over the years. And so at its outset, SAMS initially was just to have this sort of one time point snapshot of cognitive function with a focus on memory function. Mm-hmm. And we were interested in trying to understand what are some of the factors that are accounting for why somebody is learning or remembering a bit better at one moment versus another moment, as well as then between person variability in memory performance. As a function of age, but even controlling for age, what accounts for why one older adult is performing a bit better than another older adult.
Right. We know that there's this huge variability between people as we age. And so zeroing in on okay, let's look at this variability. Let's see, as you said, moment to moment, what are the things that are causing these differences or associated with these differences in performance? And what can we learn about sort of what are the factors that are influencing this broader picture of variability? Okay, that makes sense.
Absolutely. And so that was the initial idea, and we've done a tremendous amount of work there. But the exciting thing about and the privilege of being at a place like Stanford is you're surrounded by amazing colleagues. So I'm a basic sort of systems neuroscientist, cognitive neuroscientist focused on memory. What are the mechanisms of mind and brain that build memories, that allow us to retrieve or reactivate those memories? What's giving rise to this moment-to-moment fluctuation and between-person fluctuation? As SAMS began to take off and we're doing this sort of deep dive with our collaborative team, other science advances, and there are other sort of tools that one could bring online and add to the SAMS study. And SAMS as a project, and I as an individual scientist have been incredibly fortunate that Beth joined our faculty and immediately reached out when she landed on campus. And SAMS was a natural intersection for our two research interests, sort of Beth's tremendous expertise in the pathology and the imaging of the pathology related to Alzheimer's disease, along with other areas of expertise. And so over the years, from beginning in 2014, SAMS has expanded. We've added additional imaging methods, which I'm sure Beth can comment on, adding positron emission tomography with new tracers that can allow us to measure the burden of tau in individual brains and where that burden is in particular locations in individual brains, and trying to understand how variability in memory relates to that. And at the end of the first wave, we kind of thought SAMS would be a one-time point, but we've now added a longitudinal arm where we're bringing individuals back 7 to 8 years after their first enrollment. And this is actually allowing us now to answer or explore the very questions that you've surfaced with the start of this question, which is, can we try to understand which of these various factors might be drivers or predictors of change, maybe leading to cognitive decline over time, as well as try to understand mechanisms of resilience. What factors enable somebody to both maintain intact neurocognitive health, maybe perhaps in the face of some of the risk factors, some of the early pathology associated with Alzheimer's disease or other conditions. So we're excited about where SAMS is ultimately going to go in terms of being able to address these questions of resilience.
Yeah, that's so interesting. So yeah, now that you've got this 7 or so year time point, you can say not only what are some of the things that are associated with variability in people's cognitive performance as they age, but also what are the things that are associated with big change if potentially you see some people go from being 80 to 87 and you see a big decline. Some people are still on a baseline and you can start to piece apart what are the things that might account for that. In my mind, the goal seems like, how can we replicate the Harry 2 story, right? How can we all live to a ripe old age and still be able to do the things we love? So Beth, Anthony brought up the technological developments, things like PET imaging of biomarkers of Alzheimer's disease, like amyloid and tau in the brain, large-scale proteomics where we can get samples and like really see what are the differences in proteins or in gene activity and so on. I'd love to hear more examples of how some of these developments in your SAMS study and maybe elsewhere in the field are changing our understanding of the biology of how our brains age and what resilience looks like.
Yeah, absolutely. I mean, so we've, as a field, have seen an explosion of tools that we can readily apply to humans in a non-invasive way. Before this explosion, really the only way to know pathologically what was going on in individuals' brains was at death.
Right. You'd get an autopsy and have a look at the brain, and after the fact, you could see if they had amyloid in there.
Yeah, and this would be, done in the context of an individual living with dementia. So it would really just be to confirm that the dementia was due to Alzheimer's disease. So it was very limited. And now for about 20 years, so we have been able to reliably measure the amyloid protein, which we think is quite central for Alzheimer's disease. For about the last 10 years, we've been able to measure the tau protein, which is the other hallmark pathological feature of Alzheimer's disease. But maybe most excitingly, just really in the last few years, there's been very reliable, robust plasma markers of these AD proteins of amyloid and tau. So we really can get a readout of these core pathologies in a quite straightforward, easy to collect way. And we also can sample the plasma more frequently than we would do with a PET scan.
So that's just a blood draw, and then you extract the plasma and you can see signatures of whether there's amyloid or tau in the brain?
Yeah, so that's where we are as a field. I mean, there is some devil in the details. So for instance, spatial information of these proteins is probably important.
Right. Where is it in the brain? Yeah, you're not going to get that from the plasma.
We don't get that. And there's known comorbidities that might influence these plasma measures. So for instance, kidney function may push around these measurements and be less reliable. So there's some details to sort out, but in terms of a research tool, this is really an incredible opportunity to understand the consequences of these pathologies associated with Alzheimer's disease. So circling back to the normal aging question, another layer of this is that these pathologies do not appear when somebody actually has dementia. We know that they are present in some individuals decades before the onset of actual dementia. So dementia itself is defined by pretty much the opposite of Harry. It's lack of functional independence. But what we're understanding now about these diseases is that just like most health conditions, that the processes start well before symptoms. So in the case of dementia, the symptoms are overt cognitive issues and eventually loss of independence. So now what we're able to do is measure these processes in a time in which there are no symptoms. And there's many implications for this. So one interpretation is that this will give us a window into risk if you have these profiles early on. And this is what we're seeing. You're more at risk for eventual decline.
So someone who doesn't have any obvious clinical presentation, if you start seeing these signatures of amyloid and tau in the brain, that gives you a sense of maybe they're at risk of developing more clinical presentation later.
Yeah, at the group level. So on average, this is associated with a higher risk of progression later on. However, we're also seeing that there's individuals that have these signatures that do not progress. So it's another form of this variability and another potential window into mechanisms of resilience. So why do some individuals harbor this pathology that we know it likely isn't good, that it increases your risk? However, it's also possible to have this signature and remain healthy. So it provides these specific mechanistic opportunities to tease apart resilience.
I've found this so interesting. I mean, talking with folks in the Alzheimer's field for a number of years now, this comes up again and again, that so often you see someone who passes away from natural causes in their 80s or 90s, and they seem perfectly cognitively healthy. And then you look at their brain and it's full of amyloid plaques. And if someone had just looked at the brain, they might think, oh, this person probably had Alzheimer's disease, but in fact they didn't. I don't know if you happen to have these numbers offhand. Do you have any sort of statistics to give us a sense of how common that is? Is that unusual to see someone who's cognitively healthy but has lots of amyloid plaques in their brain?
Yeah, so actually this is very well studied. So amongst 70-year-olds that have no symptoms whatsoever, this includes, even mild cognitive impairment. So even excluding folks that would fall into that bucket, it's about 25 to 30% of 70-year-olds would have an amyloid signature that is consistent with Alzheimer's disease, but do not have symptoms. The patterns with tau are a little more complicated. So what we kind of believe is happening is it begins in these brainstem areas, and then it's in the medial temporal lobe focally, and then eventually spreads throughout the brain. So having this focal presentation of tau is quite common in normal older individuals. Having a very widespread tau is extremely uncommon. So that marker is better aligned to the actual clinical presentation. And that I think also gives us a hint of these mechanisms. So a potential resilience mechanism might be that, there's some folks that are experiencing this amyloid pathology, but whatever these protective mechanisms might be, in that brain, the tau is not spreading from the medial temporal lobe to cortex.
These biomarker studies that we now have, whether through imaging or through plasma, are these helping to explain differences in cognitive performance as well? I think you were saying that the tau does a bit more than the amyloid. In your research, I know you've been looking at amyloid PET and sort of risk for developing Alzheimer's disease, but does it also show sort of differences in where people are within those classifications? Like if someone has some more memory problems or is starting to slow down?
Yeah, so the way that we design our studies is when Anthony was mentioning at the beginning of the study, it was a cross-sectional look at the data, and now we're starting to look over time. So I think what you're asking is within this healthy range, there is variability, even though everybody's clinically normal. Do these markers, explain that variability? Interestingly, we have measured AD probably 5 different ways now because we have PET scans. We also do cerebrospinal fluid. We have these emerging plasma measures. So we have many measures of this one pathway. And we do see associations. If you have more of these proteins, you tend to be on the lower side within that normal range. But the total variability explained by these pathways, even though we have these very good measures, is minimal. That will probably change when we look at the data over time. So when we look at change over time, we are seeing larger effects of these proteins. But again, the percent of variability explained by these protein signatures of Alzheimer's disease, it's at most about 10% longitudinally. So I think it's telling us 2 things. One is telling us, it's important to measure these biological pathways associated with Alzheimer's disease, but it's also reminding us that we're missing a lot of the explainable variability, that we actually aren't really doing our jobs and unpacking, what's really driving this tremendous variability within our aging cohort.
If I could maybe build on that, Nick.
Yeah, please go ahead.
So that was really great, and sort of complementing what Beth just noted. The exciting thing and the powerful thing about studies like SAMS, where you are following people now over significant periods of time and with many behavioral, imaging, and other biological measures from each individual, is you can start to sort of weigh in and ask some of these questions and maybe perhaps reveal new sort of predictors of perhaps resilience or of decline. Beth already alluded to this. We're seeing, when we're looking at the SAMS participants who've already come back after their 7 to 8 year sort of period, is some of them, their cognition, their memory isn't changing at all over that 7 to 8 year period. They're remaining, as best as we can tell, cognitively healthy, neurobiologically healthy. Others are showing some decline. And so we now have this opportunity to try to understand what are some of these predictors. And as you just discussed with Beth, and Beth emphasized, sort of these amyloid and tau protein measures are sort of predictive of some of the sort of change over time. There's a new paper out just this week from our collaborators, Hamilton O, Tony Wiscori, and others, where they're looking at the CSF data from our SAMS participants, as well as from participants in other large aging samples.
So that's like cerebrospinal fluid from a spinal tap or something?
Exactly, exactly right. And they're also looking at the blood plasma. And what they reveal is that while there's some change in cognitive function that's explained by the presence of amyloid and tau, some portion of that change over 7 to 10 year period, depending upon which sample the data are coming from, a significant portion of additional independent change is coming from other proteins related to synaptic functional integrity.
Interesting.
A synaptic function that are explaining independent change or resilience over that 7 to 10, 12-year period, as well as sort of if you use imaging methods to look at sort of brain aging, change in terms of brain aging scores and things along those lines. And so it's studies like that are harnessing these different measures we have from individuals. It allows us to both test sort of leading hypotheses, like what is happening with respect to amyloid and tau, but what are some of the other unknown biological variables that might be riding together, maybe conferring some resilience, we don't know yet, but some resilience to the emergence of amyloid and tau because of these other pathways, these other mechanisms. And we, of course, as Beth noted, there's a modest percentage of that change that's explained by amyloid or tau. We're very much interested in identifying what are some of the other factors as well in terms of other systems, structural change within the brain, functional change within core systems of memory that may be occurring independent of, and it's sometimes undoubtedly interacting with these sort of AD pathways. So it is an exciting time because there's more and more of these samples. There's not a lot of them, but samples like the SAMS sort of project where we have so much data from individuals. Our SAMS participants are so generous in how much time they're dedicating to participate in the study, spending 4 days with us during the initial visit, coming back, spending 4 more days with us, allowing us to collect a tremendous amount of different kinds of measures from them. And it's these kinds of projects that are actually enabling this testing of existing hypotheses, but also doing discovery science.
I mean, it's just such a remarkable finding. You can tell me if there were hints of this earlier, but there's been such a focus on particularly amyloid and tau as well. I mean, the biology there seems fairly clear that those are involved. There are other things like inflammation and microglia and vasculature and, all these other contributors to the disease. But if you go in and look at the amyloid and tau and say, well, yeah, that affects how people's cognitive function develops as they age, but less than 10% of the difference in someone's cognitive function is determined by the level of amyloid and tau in their brains. As you've been saying, this leaves open the question of, well, what's the other 90% of contributing factors to whether someone stays cognitively healthy or goes into this decline that, I'm sure many of our listeners have seen in their loved ones as they get older. Anthony, I'd love to stick with this for a minute. What are we starting to learn about some of the variable biology of aging and resilience? I mean, what are some of the things that affect some of these other cognitive functions that allow us to maintain our independence into old age?
That's a great question, Nick. And let me maybe highlight first a few points that are principles that we've discovered in recent years or past few decades. One, if I look at the field of sort of cognitive aging and age-related change, like in memory, I would say that our sense of how much change was occurring, independent of now disease processes, how much change that was occurring, we probably overestimated it. A few decades ago, we realized, time of day when we're assessing somebody's memory behavior really kind of matters. That is, our memory function isn't static. I alluded to these sort of moment-to-moment fluctuations, and we're studying how small fluctuations in our attentional state and arousal over the course of even just seconds to tens of seconds to more sustained periods of time over the course of a day impact how well we're learning or how well we're remembering at that sort of particular moment. But we also know that there are these sort of larger time of day effects. And we discovered this a couple of decades ago that for quite a period of time, we would often, bring in our participants and assess their cognition at times that worked particularly well for the research team, who are younger adults, and later afternoon and evening often would be particularly good times. Turns out those are peak times of day for performance for younger adults on average.
Interesting.
Older adults, their peak cognition, it's earlier, in the day, sort of early mid-morning. And if you actually measure cognition in older adults and in younger adults, aligning those measurements to the peak time of day for the two groups, a significant percentage of what we thought was age-related decline in sort of true cognitive ability actually goes away.
Oh, interesting. This made me think it's so important when we read headlines about such and such leads to cognitive decline, or by the time you're this age, your cognition is going to decline so much. These are the questions that we should be asking ourselves as sort of consumers of science, which is I wonder when they measured that cognition. Is that representing what they're capable of, or is that an artifact of how the study was conducted? Is it really measuring something slightly different? So it's just one of those things where science grows over time through this basic research to understand, we need to be doing this in the morning, not in the afternoon.
Absolutely. And that gets to another fundamental and maybe a critical point for our listeners to appreciate, which is we live in a culture, we understand what tends to happen to humans through time. And one thing that we have associated with aging is cognitive decline. And as we've discussed, many older adults do not show cognitive decline. That stereotype, and with respect to at least particular diseases like Alzheimer's disease, one kind of cognitive decline is memory decline, creates a lot of potential psychological processes for an individual that can lead to underperformance for that individual, not because they actually have a true decline, but because they're worried about this stereotype about being an older adult. Maybe my memory might be changing. If my memory changes, maybe it's a marker of, maybe I'm in this early stage of slipping in towards Alzheimer's disease. That induces that psychological threat. So what my colleague Claude Steele has described as sort of stereotype threat. That threat induces an anxiety response that we know actually degrades performance,
right,
and can actually lead to underperformance. And there's one study out there that you bring older adults in and you give them a standard cognitive task that's used on the front end in clinical assessment to see if there's in fact some cognitive decline. And you sort of prime that age-related stereotype that cognition might decline with age, versus you don't prime it, and a significant percentage more of older adults underperform on that task, leading to scores that might have you a little bit clinically worried about how well they're doing relative to if the stereotype isn't primed. And so this makes this just higher level point, which is what you want is a readout of what is your true sort of capability, your true function, and that it's hard to get from a single measure. You can't really have confidence that you kind of know what your ability is. You might underperform because you didn't sleep well that night. You might be stressed because you're worried about your performance. And that could then create further worry that you might be showing cognitive change.
Yeah.
It's a challenge for the field. It's a challenge for individuals when they think about, they're trying to think about their own cognition.
Yeah. And I remember, I think you told me that, some of these results that Beth was telling us about with being able to see using PET imaging, that some people actually have subclinical levels of, or maybe preclinical levels of amyloid and tau in their brains, might say that, well, there are people who we've thought were quote unquote healthy aging, and their performance is actually lower because they have preclinical Alzheimer's. It was not clinically diagnosed, but they've got amyloid, they've got tau in their brains. And so that also might be lowering sort of the expectation for the average person as they age. Is there anything else that you'd want to say about that? I thought that was a really interesting point.
So I will, and this builds on Beth's earlier comment about how much variance is explained by these variables, right? And so it's clear that it's really likely when we're trying to look at what happens with age in individuals who are, as best as we can tell, cognitively unimpaired. We are enrolling these older adults who are cognitively unimpaired largely by assaying their cognition, their cognitive behavior. But as Beth noted, maybe 25% of them have elevated amyloid levels. A subset of those individuals have elevated tau. And in the SAM study, in the literature, some of the differences between healthy older adults or putatively healthy older adults relative to younger adults in terms of memory performance is likely due to those older adults who happen to be at this early stage, perhaps, of Alzheimer's disease. And it's a small amount, as Beth noted, and this is an important point. It's a small amount of that variance, right?
Right.
Because these are cross-sectional studies rather than longitudinal studies. But I do suspect the next direction, this is the direction we're taking in our work, is to not just, if you believe you're trying to understand cognitive and neural changes in individuals who you believe are not in the earliest stages of Alzheimer's disease, the strategy is going to be a combination of behavioral assay as well as probably blood plasma assay, so that you can bring in individuals who appear to be cognitively healthy and who, as best from these blood plasma measures, appear to have low burdens in amyloid and tau.
I want to make one comment on this too.
Sure, please.
In terms of kind of expectations for aging and kind of what we're learning from SAMS about like decline in general. So I think, what we've clearly learned is that it is certainly possible to remain cognitively stable, and actually after a decent follow-up time too. But one, I think, criticism of our work is that we've really designed these studies to really get at very healthy aging. And what I mean by that is our cohorts are not representative. When we enroll our participants, if they have significant comorbidities, medical conditions, they're excluded. If they have implants in their body that preclude MRI scanning, they're excluded. If they have complaints about their cognition, they're excluded. And the reason we do this is because we're trying to get at healthy, optimal aging. And it's interesting, even with this very healthy sample, we still find, evidence of early Alzheimer's, et cetera. We also, we have our participants come for 4 visits to Stanford. They are just very committed, and I think represent another kind of sign of how healthy they are, the ability to do, 3 imaging sessions and a lumbar puncture and blood draws. So we have to be a little cautious in interpreting these results as this is what happens to everybody in the world as they age. Because it could be possible that if we actually really did a random sample of 70-year-olds, we may or may not see more decline. I would suspect that we would.
Right. I mean, how many of us get to go through our 70s, 80s, 90s without major surgery or some other life event that is going to add a lot of stress or cause, some other insult that would probably preclude someone from being in your study. Not that those specific things are necessarily your determining factors. It reminds me a little bit of, we have a story that should have come out by the time this airs, which we'll link to in the show notes, about your colleague Martin Angst doing this long-term study of people post-surgery that's supported by the Knight Iniative to see, well, we know that after a major surgery, a lot of older adults do go into a decline following that surgery for one reason or another. And so trying to follow people long-term and say, what are the factors, again, that determine whether someone is resilient to that stress or succumbs to whatever is going on, that can be the trigger that leads to the decline. It reminded me, there's also this thing that I think a lot of people probably notice in their friends and family members, that aging is not a linear process. It's not like we age the same amount every year. The Snyder Lab came out with a paper last year talking about aging spurts that got a lot of attention. And, there's something going on biologically that we age in jumps. And I'm sure everyone knows someone in their family, and you can see, like, maybe you don't see them for a year and you come back and you see, oh, wow, they've really aged. Do we know anything about the biology of why someone might suddenly change their cognitive performance over the matter of a few years?
So for neurodegenerative diseases, when that process is actually initiated, even though there's potentially a long asymptomatic phase, and then maybe some subtle changes that are very difficult to dissociate from kind of healthy changes, that's kind of linking back to Anthony's kind of conversation about the difficulties with measuring kind of true performance. So there's clearly this overlapping phase, but when somebody is actually progressing on this dementia cascade, there is going to be what might seem like out of nowhere, a sharp, rapid decline. And I mean, that's kind of the characteristic feature behaviorally of somebody as they're entering that AD dementia phase. And we also know that kind of follows from what we know about the tau pathology as well. So I mentioned earlier that it's very uncommon to see distributed tau tangles throughout the brain in healthy older folks. If we see some tau signal with PET, it's usually quite focal within the medial temporal lobe. And what we know from these longitudinal studies is that it does seem like there potentially is some sort of trigger or spurt event where this pathology leaves the medial temporal lobe and spreads throughout the brain. And that trigger seems to be quite important for the actual clinical expression of Alzheimer's disease. So I think that, is potentially one example of a process that might be, a little bit linear, but then an event happens and then that person truly experiences decline that leaves this range of ambiguity and difficulty in terms of measurement.
Well, to close us out, I'd love to go back to this idea of how do we live the dream of resilient aging? How can we all live like Harry II, who we talked about at the top of the episode? Are there any patterns that you're observing in the resilient aging cohort in the Stanford Aging and Memory Study or in the literature more broadly that can teach us something about the factors that are in our control or that we might be able to intervene and improve our chances of living and independently into our 90s?
First, so there's constant discoveries happening, right? There's a paper out just this week about unexpected benefits of vaccines for shingles. Mm-hmm. And so as we continue to accumulate more and more sort of data from individuals, and we follow individuals, so, larger and larger groups of individuals over time, I suspect there will be more and more discovery. We, of course, know many of the things that, or some of the things that matter in terms of remaining socially engaged, remaining cardiovascularly fit, remaining cognitively engaged, sort of have a healthy diet, et cetera, that seem to have some modest protective benefits and might increase the probability of you remaining resilient and sort of having a very long neurocognitive health span. And that's what we want, right? We want to stretch health span to align with lifespan so that this spurt of maybe rapid decline that occurs may occur only at the very end of life and that we're healthy up until that particular moment. Yeah. We're also discovering, and I can't say that I have anything specific to comment on here, but I did want to sort of weigh in. There's other discovery science that's occurring over these same kinds of measures that we're collecting in SAMS, including these CSF and plasma measures, where we're not only able to track these proteins related to, say, Alzheimer's disease, or now with this new paper, potential resilience over a 7 to 10 year period, you can also identify proteins that are differentially associated with aging of different, organs. Not just the brain, but other proteins that are associated with aging of other organs. So organs aren't all aging at the same rate, and the brain is not immune to what's happening in the rest of the body. And as we go through time, we may actually find out that there's interactions, and some of these sort of spurts or changes in brain age might be coming about through interactions with aging and age-related processes or disease-related processes in other systems. So there are things that we think if you were going to give guidance to, somebody, to a loved one, to a friend, stay neurovascularly fit, remain engaged socially, cognitively, have a good healthy diet, et cetera. But there's a lot of things that we just don't know. And I'll turn to Beth on this in just a moment, but I want to take this moment to not only sort of express my appreciation for the support we've received from the National Institute on Aging, that's enabled SAMS and related studies, as well as sort of the Tsai Neurosciences Institute, but also the tremendous generosity of our participants in SAMS and the other aging studies that we're conducting. Because it's through this support, both federally, privately, as well as from our participants, that we're able to actually engage in this discovery science with the hope that we ultimately will have even more deliverables, more things we can say in response to this question about how can we stretch our health span to the latest periods of our lives.
That's such an important point, Anthony. We need to stay curious and keep investing in understanding the fundamentals if we ever want to use these insights to improve human health and well-being. Beth, was there anything that you wanted to add to that?
Yeah, I guess I would just add, I agree with everything, Anthony, you just listed, and the importance of emphasizing the lifestyle choices we have. I do think, anything that's good for the heart is good for the brain, it's good for all your organs. It also seems there's, evidence that even strength training and things like this and at older ages could be very beneficial. So I think we have a better understanding of these kind of exercise-related mechanisms too. But I think, unfortunately, we don't know what the profiles and the factors are that predict this sort of resilience. And it probably is quite heterogeneous. There's probably, based on somebody's genetics and interactions with the environment and their lifestyle, it probably is quite complex. So in a sense, I do think these longitudinal studies are so critical to continue, and the deep phenotyping approaches, that's the only way we're going to have concrete answers to these questions, is to continue doing this work.
Right. There may not be one secret sauce or fountain of youth, but remembering that it is possible, right? Humans can live into their 90s and past 100 with their cognitive function intact. And in fact, probably more people do than we recognize either in the scientific literature or in our lives. Like, maybe we can all get there.
Yeah, it's a kind of a reverse engineer problem that we have. So we definitely, we find the examples, quite easily. So we know it's possible.
Mm-hmm.
And what's great is it really feels like we're on the cusp of real breakthroughs. The rate of discovery over the last sort of 10, 15 years and the new methods we have, it really feels like we're on the cusp of figuring out some of the other critical processes that are going to essentially enable us to be resilient.
Fantastic. Well, that's a perfect place to end. Thank you both so much for coming on the show and telling us about the cusp of discovery.
Thanks, Nick. That was fun.
Yeah, thanks, Nick.
Thanks again so much to our guests, Beth Mormino and Anthony Wagner. Beth is an associate professor of neurology and neurological sciences here at Stanford, and Anthony is a professor in the Department of Psychology and one of the deputy directors of the Tsai Neurosciences Institute. You can read more about their work in the show notes. If you're enjoying the show, please subscribe and share with your friends. That's how we grow as a show and bring more listeners to the frontiers of neuroscience. We'd also love to hear from you. Send us a note at neuronspodcast@stanford.edu with your thoughts on the show, suggestions for research topics, or just generally to say hello. From Our Neurons to Yours is produced by Michael Osborne at 14th Street Studios with sound design by Morgan Honaker. I'm Nicholas Weiler. Until next time.